write some comments at the end. So today I have the honor of introducing someone many of you know. Dr. Wisniewski is a board-certified neurologist and neuropathologist. He holds a number of positions here at NYU, including director of the NYU NIH-funded Alzheimer's Disease Research Center, the Pearl Barlow Memory Disorder Center, the Conformational Disorders Lab, the Neuropathology Fellowship Program, as well as the director of the Center for Cognitive Neurology. In addition, he's also professor in the departments of neurology, pathology, and psychiatry. Dr. Wisniewski has written over 300 peer-reviewed publications and holds 23 U.S. patents. He's been continuously funded by the NIH for over 25 years, with much of his research focused on developing novel therapeutic methods for Alzheimer's disease. Today, he will be presenting on one of the frequently asked questions, will there be a vaccine for Alzheimer's disease? So with great pleasure, I'd like to introduce Dr. Wisniewski.
Hi, everyone. Thanks very much for that nice introduction, Ashley. Today I'll be speaking about a topic that's near and dear to my heart, the development of immunotherapeutic approaches like vaccines for Alzheimer's disease. And certainly this is an exciting time with many developments in this area. So what we'll be going through in this seminar is listed here. And I'll go through each of these topics in turn. So in terms of the major causes of dementia, there are many such causes. Certainly Alzheimer's disease at the center of the slide is the most common cause, but there are other major causes like vascular dementia, which can be due to multiple strokes. And there are many other neurodegenerative conditions like Lewy body dementia or frontotemporal dementia, which can also cause a dementia. And that's why it's important to come to specialist medical centers like the Barlow to get an accurate diagnosis, because each of these conditions has a different prognosis, and the therapeutic approaches are distinct for each of these entities, but they all present looking like a dementia. Alzheimer's disease is by far the most common of these causes of dementia and is in fact the sixth leading cause of death. It's extremely common in the USA. There's almost 6 million people affected, and almost every minute someone in the US develops the disease. The cost associated with taking care of patients with Alzheimer's disease is staggering, over $300 billion this year. And these costs are expected to rise exponentially to a level that could potentially bankrupt healthcare in the United States unless interventions are developed, which hopefully they will be. So if we look at how medicine in general is doing against some major killers, looking at changes since 2000 compared to 2018, we've done well for many major problems like heart disease and stroke with significant declines. But unfortunately, Alzheimer's disease is the only cause of death among the top 10 in the USA, where we really have no effective means to prevent or cure this disorder. And as a quick aside, in this COVID pandemic, people with Alzheimer's disease and related dementias are particularly vulnerable to severe COVID infection, and indeed their presentation can be neurologic, distinct from the usual COVID presentations. And we recently published our own experience here locally in the first wave of COVID at NYU, where we had over 5,000 inpatients with COVID, and many had neurological complications. So this is an issue that is affecting this population greatly, and it is something that we're very much involved with trying to treat and prevent here at NYU. The major lesions of Alzheimer's disease are listed here: the amyloid plaques, neurofibrillary tangles, vascular amyloid, and all of that leads to neuronal and synaptic loss. Amyloid plaques are shown here in this micrograph of the human brain, looking at the gray matter of the brain, and it's stained for amyloid deposits using antibodies to the protein that gets deposited in amyloid beta, in these amyloid plaques, amyloid beta, highlighted with the arrows. And these deposits are extracellular, and the brain becomes full of these toxic plaques. These plaques can also be seen using different methods like silver stains, and you can see in this gray matter here, there's many of these dark gray and granular blobs, which are amyloid plaques picked up by the silver stain, and this really causes brain dysfunction. Neurofibrillary tangles are these accumulations of the tau protein, abnormally phosphorylated tau protein in neurons, and this causes dysfunction of the neurons, and the neurons being the major cells in the brain that we think with. So this causes cognitive loss. And again, with the silver stain in a section of the hippocampus of the human brain, you're seeing most of the neurons here being filled with abnormally phosphorylated tau in the shape of these flame-like neurofibrillary tangles that accumulate inside neurons. Another major pathology is the accumulation of amyloid beta in blood vessels in the brain, which is what you're seeing here in this circular blood vessel, and also in the covering of the brain and the blood vessels around the brain, and the leptomeninges, that's the covering of the brain. So this is another type of pathology that leads to disturbances of the blood-brain barrier and blood flow to the brain. So grossly, this is a section through the normal brain, but as Alzheimer's develops, the first thing that occurs is shrinkage of the hippocampus, and that's the part of the brain here that we have our short-term memory stores, and this leads to problems with encoding and retrieving memory. And then as the disease progresses, there's a little bit of more generalized cortical shrinkage. And then ultimately, as the disease progresses, the hippocampus almost disappears completely, and the fluid-filled spaces in the brain become enlarged, and there's clearly much more generalized shrinkage of the brain, with also enlargement of the lateral ventricles. The basic pathology here is tied to these two proteins, the normal amyloid beta peptide and the tau protein. Both of these are normal proteins with specific normal functions in the brain. In the setting of Alzheimer's disease, both of these proteins form clumps, and form these soluble oligomeric structures that float around in the brain, and then ultimately can form large aggregates that precipitate in fibril form, with the amyloid beta forming the amyloid plaques and the conchophilic amyloid angiopathy, and the tau oligomers forming neurofibrillary tangles. It's the soluble oligomeric species of both amyloid beta and tau that are the most toxic to the brain and are most linked to neuronal loss. This is a process that occurs over very long periods. So in this graph, you see the preclinical period of Alzheimer's disease extends decades until one develops mild cognitive impairment and ultimately dementia. The amyloid beta accumulates first over a period of some 25 years before you have the development of mild cognitive impairment. And the accumulation of amyloid beta is followed by the accumulation of the tau pathology. And this can be picked up using biomarker studies like amyloid PET and changes in the cerebrospinal fluid with reductions of A beta 42 as it gets deposited and increases of the tau protein in the cerebrospinal fluid, and then ultimately also with structural MRI, where one can see that brain shrinkage that I highlighted in the previous slide. So there's a big move in our own center and in other Alzheimer's centers to diagnose patients very early in this process when therapeutic interventions are perhaps most likely to prevent the progression to MCI and ultimately dementia. Alzheimer's disease is broadly divided into early onset familial forms, which are less common, account for less than 5%, and the more common sporadic late onset, which occurs after the age of 65, but in most folks above the age of 75. And in the early onset forms, genetics plays a big role with mutations in these particular genes that can drive the production of amyloid beta. In late onset Alzheimer's disease, the picture is more complicated with many environmental factors increasing the risk of developing Alzheimer's disease, but there are also a number of genetic risk factors. And the genetics here are quite complicated. So in the early onset forms, as I mentioned, mutations in these three genes can be really driving amyloid beta production. But in many recent studies, genetic studies, there are many genes that can drive late onset Alzheimer's disease. And these genes interact with environmental factors like head trauma and the vascular risk factors. So it's a complicated situation. However, the most important genetic risk factor for late onset Alzheimer's disease is related to apolipoprotein E isotypes. So in the general population, there are three common ApoE isoforms, E3, E4, and E2. In late onset Alzheimer's disease, there's a big overrepresentation of ApoE4, and having this ApoE allele is something that it drives Alzheimer's disease pathology, whereas having the ApoE2 allele is protective. So if one has one copy of E4, it increases the risk of Alzheimer's disease 2 to 3-fold. But if you have two copies of ApoE4, it's quite a dramatic increased risk, up to some 10-fold. So this is the amyloid cascade hypothesis of the causation of Alzheimer's disease. So under normal circumstances, there's production of monomeric single A beta peptides throughout the fluids in the body, and that's normal. But in Alzheimer's disease, this A beta peptide aggregates and forms these toxic oligomers of A beta and ultimately fibrils that deposit in the blood vessels in the brain with the congophilic amyloid angiopathy, and in plaques in the gray matter. All of this is toxic, and it leads to the production of neurofibrillary tangles, these accumulations of abnormally phosphorylated tau protein, and all of that drives neuronal toxicity and ultimately death. the presence of the ApoE4 allele, in fact, promotes this aggregation of A beta and the formation of these toxic oligomeric species. So that's really what we think is the causation of Alzheimer's disease. And again, here is this amyloid cascade hypothesis shown on the left-hand part of the slide. And in terms of interventions, we certainly have symptomatic drugs like Aricept, which is an anticholinesterase inhibitor, and Namenda, which is an NMDA antagonist. But these medications, although certainly useful, are mainly symptomatic drugs. However, there are many things in development which are really much more disease altering. And these approaches, in particular, immunotherapeutic approaches, different types of vaccines that are showing great promise. So there are many ways of manipulating the immune system to try to reduce or prevent Alzheimer's disease pathology. So one can either induce the immune system to produce antibodies or directly inject antibodies with passive immunization that are directed to the A beta pathology in Alzheimer's disease. One can do the same thing for the tau-related pathology here. And there are also some approaches where one can induce an immune response that targets both of these toxicities in Alzheimer's disease concurrently. And that's something that we in particular in my lab are involved with. And that approach of inducing an immune response to both A beta and tau pathology is something that we've developed in the lab and have studied preclinically. And that approach is summarized in this slide. And what we did is study a very rare form of dementia called British dementia, where an amyloid protein is deposited in the brain, but that's distinct from amyloid beta and tau. However, this protein also produces toxic oligomers, and using that shape of toxic oligomers, we developed active immunization and passive immunization approaches that can induce the immune system to attack both the A beta and tau pathology at the same time. So we've used many mouse models of Alzheimer's disease with this approach and showed that using various mazes, doing active and passive immunization with these P-BRIE approaches leads to major cognitive benefits. And that's shown in this graph here, where the control transgenic mice make lots and lots of errors. These mice are demented, so in these mazes, they can't find their way, whereas the Alzheimer's mice that receive this P-BRIE treatment, they perform with far fewer errors, and they in fact perform like normal mice. And this is very complicated slide summarizing this approach, but this approach has been put forward in a number of publications, as well as issued US patents that fully explain how researchers can reproduce our findings and use this approach to treat Alzheimer's disease. So just to illustrate what one of the more recent experiments we did with these Alzheimer's disease mice that are demented, we injected monoclonal antibodies, antibodies that are directed to the oligomers of A beta and tau, into these old demented mice. And we showed that with this treatment, the pathology that they developed, and these are all these brown lesions, and this is the mouse hippocampus. So this is a control mouse where there's lots and lots of Alzheimer's pathology as highlighted by these brown deposits. When we treated the mice, you can see a major reduction in the amount of amyloid pathology in the treated mice, and this correlated with big cognitive benefits in these animals. We are also developing many other antibody-like approaches. So in addition to inducing the immune system to produce antibodies or develop monoclonal antibodies, one can engineer antibody-like proteins. So these are antibody mimetics. So they're similar to regular antibodies. And one of these are Affibodies. And Affibodies are already used clinically in some cancer treatments and some diagnostic treatments. We also engineered our own Affibodies, and we produced Affibodies that bind to the amyloid beta protein and use them to treat the demented Alzheimer's disease mice. And we showed using our particular engineered Affibodies that with treatment, the demented mice, in fact, had big improvements and they performed similar to wild-type mice, whereas, as shown in black, the control Alzheimer's mice made lots and lots of errors. So we're using this approach to engineer other Affibodies that could be potentially used for Alzheimer's disease treatment, as well as the causes of other dementias like Lewy body dementia and frontotemporal dementia. Another approach that we're using is to mobilize the innate immune system, which is a very important part of immunity in our bodies. One way of stimulating the innate immune system is by using what's called CpG oligonucleotides. And these are DNA fragments that are similar to bacterial DNA, and they can stimulate the immune cells in our body. And this type of stimulation with CpG ODN is already being used to help in things like cancer therapies and in vaccines. In fact, it's being used to develop COVID vaccines currently. So we took this approach of using CpG ODN in Alzheimer's disease model animals, and in two different models here, we're showing reductions in the CpG ODN treated animals with major reductions at both the A-beta, the amyloid beta pathology, and also the tau, the neurofibrillary tangle pathology, in multiple mouse models. We've since gone on to another Alzheimer's disease model, but this is a natural aging model of squirrel monkeys. And squirrel monkeys develop Alzheimer's disease pathology as they get older, and they also develop dementia as they get older as well. And in these slides of the monkey brain, you're seeing these black deposits and also highlighted in green here, and this is amyloid beta in plaques and congophilic amyloid angiopathy that these aged monkeys spontaneously develop. We've developed various cognitive tests in these animals where, as shown in these pictures, they have to find a grape reward underneath a bucket, and there's only one way of getting to that grape, and that's what the monkeys have to figure out. And old demented monkeys do worse than young monkeys on these tests. However, with the CpG treatment, the old monkeys perform much better, similar to the young monkeys. So in effect, the treatment sort of rejuvenates their brain. And this correlates with big reductions in the amyloid deposits in the CpG ODN monkeys, as shown here on the left, as compared to on the right. And this is all quantitated. So these are big reductions in the amyloid beta deposits of these aged monkeys of around 30 to 50%. So these results in both the mouse models as well as in the monkeys are much more similar to us. We're looking to take this to patient testing sometime early next year. However, in the Alzheimer's community as a whole, and in the research studies that are supported by the National Institutes of Health, specifically the National Institute of Aging, there are many, many clinical trials that are ongoing, and they're targeting many different aspects of Alzheimer's related pathology. So there's many approaches that are being tried right now, with over 230 such supported trials. So there's lots of different ways that researchers are trying to address this problem and develop useful therapeutic approaches. And that's certainly something that's available to patients here at NYU through the Barlow Center, where accurate diagnosis is made and a treatment plan. And for patients who are interested and are appropriate, they can be referred to the various clinical trials that we have ongoing. And just to highlight a few of those, we have clinical trials of the potential benefits of doing exercise. There are observational clinical trials where we do brain imaging. There's a clinical trial of testing an antiviral agent, valacyclovir, in the setting of Alzheimer's disease, addressing the viral hypothesis of Alzheimer's disease. And we have a number of antibody-type treatments that are being tested with antibodies to those aggregated and toxic forms of amyloid beta that are being tested. An approach that's generated lots of excitement, and we are also involved in this clinical trial here, is with an antibody called aducanumab, which was generated by a company called Biogen. And what was done here is that researchers looked at naturally occurring antibodies in elderly individuals that were directed to aggregated forms of amyloid beta. So some people naturally produce antibodies to these toxic forms of amyloid beta. And what researchers did is isolate the lymphocytes that produce these antibodies in patients naturally, and using various recombinant approaches actually produced monoclonal antibodies that could be then tested in Alzheimer's model mice. And what was shown is that these monoclonal antibodies would bind to aggregated A beta in the plaques in the brain of these mice and induce clearance by macrophages. So the macrophages are the cells in the brain that take up the garbage. They clear out the garbage, or they destroy invading pathogens in the brain. So in this way, the Alzheimer's related pathology can be cleared. So this antibody, the aducanumab, has been subjected to recent phase 3 trials, which were the EMERGE and ENGAGE trial. And as all clinical trials, there was a placebo arm, but there was also 2 different doses of aducanumab that were given, which were low dose of 3 and 6 milligrams and a high dose of 10 milligrams. And in folks that were part of the EMERGE trial, there were significant cognitive benefits that were produced, and these were measured by a cognitive test called CDR sum of boxes. And as you can see in particular in folks that got the higher dose, the 10 milligrams per kilogram of the aducanumab, there were substantial benefits with reductions in the cognitive loss that was quite significant, as shown in this graph. And then looking at the biomarkers of Alzheimer's disease, the deposition of amyloid beta in the brain as measured by amyloid PET, there were reductions in the amount of amyloid in the treated patients that was dose dependent. So the folks that got the higher dose had bigger reductions, whereas the intermediate also had reductions, but they were less profound. And in the red line, you're seeing that actually in the control group, there's a slight increase in the amount of amyloid beta. And importantly, also for the tau-related biomarkers in the cerebrospinal fluid, treatment was associated with reductions of this p-tau in the CSF. That was, again, dependent on the dose. So people that got the higher dose of aducanumab had bigger reductions. This is very promising data showing really for the first time very definitive cognitive benefits with an immunotherapy, an antibody to aggregated and highly toxic forms of amyloid beta. There were complications that were seen in this clinical trial, specifically these amyloid-related imaging abnormalities or ARIA. However, in the vast majority of cases, these side effects were quite minor and very manageable. And in fact, very soon, just next month, the US FDA will be considering the possibility of approval of aducanumab as a disease-modifying therapy for Alzheimer's disease. So there's the potential of a new therapeutic approach very soon, in the beginning of next year. An exciting time. So certainly we have many clinical trials ongoing at NYU, but also there are many clinical trials going on throughout the United States. And one can find information about these various clinical trials at these websites, which are very good, the Alzheimer's Association website and the NIA and clinicaltrials.gov. But also we have a local clinical trial team here that can be contacted for information. I think the future for a potential Alzheimer's disease vaccine is looking at the moment fairly bright, and there is a possibility that we're coming to the beginnings of more disease-modifying therapeutic approaches for Alzheimer's disease. And on the research side, we certainly hope that that's something that's going to pan out. And in happier pre-distancing times, we send you greetings from the ADRC team. And we hope we'll be able to gather again like this in the not too distant future in the coming months. But thank you for listening to this presentation.
And thank you again, Dr. Wisniewski. That was extreme.